Yes, so dogs with CLN2 or late infantile Batten disease exhibit similar signs to kids with the disease. They have an early onset of retinal degeneration. We can assess that with electroretinography or recording the electrocoil spots from the retina in response to a flash of light. They exhibit thinning of the retina and loss of retinal cells, and we see that in both in the dogs and in the children as vision loss progressing to blindness. In the past, we've had a lot of success treating the brain by delivering the enzyme to that tissue, so we thought that if we could deliver the enzyme to the retina as well, that we could prevent vision loss. So the way that we did that was to isolate mesenchymal stem cells from the dog's bone marrow. We do that so that the cells will be from the dog and not seen as foreign. But then we modified the cells in culture to produce the enzyme that's deficient in this disease, and then implanted the cells back into the eye, where we take advantage of cross-correction, the idea that the cells will produce the enzyme and that it can be trafficked to cells in the retina through normal processes to perform its normal function. And we hope that that would prevent the retinal degeneration that occurs. And indeed, we saw that. We saw a 3-month delay in the onset of function deficits in the retina, and we saw a benefit throughout the entire disease course when compared to untreated dogs. We also saw an improvement in retinal structure. The retina in diseased dogs is greatly thinned by the end of the disease course, and we saw a normal retinal thickness, just indicating that all of the cells are still there, and so we can correlate what we saw functionally with what we see structurally, and basically know that we have a healthy retinal tissue. In a human, you can say, can you see this? So we can't really do that in dogs, although we are developing some tests to evaluate so-called functional vision. But we can look at the response of the retinal cells to light, and that's done with electroretinography. We basically have a stimulator that we set up in front of the dog's eye. It flashes a light into their eye, and we have a little contact lens that we put on the cornea, and it records the retinal response to that flash of light. And then the size of the response tells you how well the cells are functioning. So we can do that over time in the dogs, and we know based on previous work that that signal is drastically reduced throughout the disease course to where it's almost non-recordable at end-stage disease. But by treating the retina with TTP1, the enzyme that's deficient, we were able to greatly preserve that response. Next steps, we're optimizing dosing. So we saw a 3-month delay in the onset of deficits, but then we start to see a waning effect. So we're considering a second injection. Right now, we've had a lot of promising results with a single injection, but we think maybe if we treat about halfway through the disease course with another injection, that we can maintain a normal retinal response. And so we're working on that. We're also considering using them to treat the brain in this disease. With a similar mechanism, we would inject the cells into the cerebrospinal fluid that circulates throughout the brain, with the idea that they would again deliver the enzyme, and it could be transported between cells in the brain.